Hello, John McGrady here again in my Interviews with Statisticians series, and today is a landmark day. It's actually the first time we've had more than one guest on the show, and I'm thrilled to have this group here from the SMART Technology Group, which stands for Statistical Methods in Research and Technology. And I will introduce the members that I have interviewing here. First up, we have Associate Professor in the Department of Biostatistics, Ciprian Crinicianu. And he's actually done a lot of groundbreaking work in the world of trying to estimate, in regression context, nonlinear associations when you have complex data structures. So he's worked in mixed models methods for extending linear and other types of regressions, and also smoothing spline methodology. And currently he's taken that and other skills to the framework of working on imaging data and movable data collection systems for like accelerometers, etc. And so today we'll be talking about the data imaging part and his work with SMART. So welcome, Ciprian.
Thank you.
We also have Elizabeth Sweeney, who's an SCM graduate from the Department of Biostatistics and currently working closely with Ciprian on issues of predicting lesions using longitudinal brain imaging data, and she's going to demonstrate verbally how that works, and we'll show some images to go along with it. And she'll tie it into stuff that we actually start with in this class, logistic regression. So welcome, Elizabeth.
Thank you.
And then we have John Micheli here, who's actually a PhD student in the department who also did his SCM as well. And he's been working closely with Elizabeth and Ciprian. And he's actually been leading the charge on how to visualize complex imaging data, which we'll describe a little bit about the data structure, but you have multiple images taken on an entity like a brain at different points in time. And he's worked on pulling those different time scans together into a user modifiable imaging presentation that helps the researcher and clinicians actually make sense of the data in visual format, and he'll be talking about that and showing a demo. So I'm really actually thrilled to have all three of them here. What they're doing is really exciting and groundbreaking, and we'll get to see some cool pictures on top of it. So let me start with Ciprian. So Ciprian, you know, you've been involved in this kind of work for the past couple of years. You guys have made some really cool discoveries in how to handle imaging data, etc. So can you tell me a little bit about what you've done so far and where you see this field going, and what are the challenges and opportunities both statistically and computing-wise? So,
thank you, John, and it's really a pleasure to be here, and we are both, all of us are thrilled to be here. And about 2 or 3 years ago, we started a lot of work on brain imaging, specifically MRI and CT imaging with applications to multiple sclerosis. This is a very big study that collaborators from the National Institute of Health came to us with somewhere around 200 to 300 subjects observed over 10-15 years with multiple sclerosis. And one of the main problems there is to try to find where are these lesions that appear in multiple sclerosis in the brain, and find them in an automatic way, find them fast, because currently they are just obtained using a manual segmentation. So people go in and then manually segment them, which takes an enormous amount of time.
and introduces biases.
Introduces biases. It's unclear whether or not they are correct. So we try to understand this as more of a data level, where we look at the images but understand the actual data and try to translate it into maps, and how these maps evolve over time. More specifically, we're interested in, say, a person has multiple sclerosis and they are seen over multiple visits, maybe over 10 years. We would like to essentially identify what lesions appear when and how. So this is the major component of.
So is it sort of the natural progression of the disease?
The natural progression of the disease in the brain.
How it impacts the brain.
And then we realized in the work with John that a similar problem appears in other types of diseases, as for example in stroke, where lesions are actually strokes, and they happen in a different way, and they tend to be much bigger and have different type of shapes and sizes. But we would like to understand from a statistical perspective how to even approach the problem. So this requires a lot of exploratory data analysis, requires a lot of understanding the problem, requires a lot of looking at the data and kind of going back and forth, sometimes in a collaborative environment as we have, and learning this as we go.
So let me just put in a plug for looking at the data. You guys hear it here, and you've heard it over and over again. That don't be fooled, the methods we use can get complicated and sexy and sophisticated, but they really, all things start with looking at your data. It's a very important part of any analysis.
Yeah, so the one thing that I would like to add is that I think that this research, I mean, I'm leading the group, but this research in particular was done by, was started by Elizabeth, who had an internship at NIH. And we were looking at complicated approaches and machine learning techniques and all sorts of things to try to predict this. And I think that one day Elizabeth and Taki Shinohara, a former student of ours, came to me and said, I think we can do this much simpler using, you know, a logistic regression. And I said, maybe we should try that. Maybe we should see whether the simple, easy to understand things works as well or better than the other things. So this is how everything started.
Excellent. So you sort of went back to basics and sort of
went back to basics.
Well, let me ask you just about the exploratory data process, though, and I'll get to John as well to talk about this. But, you know, it's a little more complicated in this kind of situation because of the amount of data you've got, right? So even before you get to the models and the logistic regressions and such, even actually just looking at pictures as such as simple box plots and stuff takes a fair amount of pre-processing and reduction, right?
Yeah. So absolutely. I mean, this is one of the first challenges. The first one is that the amount of data, right? You can have millions of observations within a brain for a subject at one visit. But that's not the only problem. The other problem is that we didn't realize that the images themselves are expressed in arbitrary units. So you need to actually standardize them and make sure that, first of all, the same location in 5 brains means the same thing, and which would be what we call spatial registration. And you also need to make sure that the numbers themselves, the gray levels in the image, mean the same thing, which would be more intensity normalization and registration. So all of these are very hard problems, and we had to address them in a way or another, sometimes imperfectly, but sometimes,
just to get started even.
We are happy with the 95% solution sometimes.
Excellent. And so what do you mean by that? That most of your good results come from basic principles, or?
Yeah. So I think that in the end, you know, going back to basics, understanding, you know, the basics of simple things, like when you do a regression and you look at the same X, the same variable across subjects, doesn't mean the same thing. We take it for granted in epidemiological studies, where
age always means age, right?
Age always means age, gender always means the same thing, but when you go to a brain image, you just have shades of gray, and that's, the shades of gray are not the same across subjects.
Interesting. So gray doesn't mean the same thing.
Exactly.
Interesting. Well, on this data subject, actually, let's bring Elizabeth into the fold, because she's worked with this registration process and sort of taking the data from multiple people at multiple time points and then filtering it through the algorithm to use longitudinal data within one person to detect lesions, and also comparing across different people to see who's more likely to have lesions where. So, Elizabeth, could you tell us a little bit about the structure of this data, and we'll show some slides to accompany her excellent verbal descriptions, and then she'll get to culminating in talking about this logistic regression procedure she uses to predict lesions and how that works.
Definitely. Thank you, John. I think I'll start by just going over the problem that Ciprian already described, but we have a great slide to help visualize that. So we've got MRI scans from 2 time periods. So we have a baseline scan and then a scan at a second time period. And we want to see what's changed in these scans. So these patients have multiple sclerosis, and we want to see where are their new lesions and where are lesions getting bigger. And so the typical way that this is done, that Ciprian already mentioned, is that people will typically go in by hand and they will kind of mark where these lesions are. But that takes a really long time to go through an entire image of a brain and find all of these new lesions. And it can also be quite variable depending on the person who's doing it, and even within a person if they do it.
So the inter-rater agreement, even within different time frames, is not necessarily good.
No, not yet.
So
and sometimes we actually found new lesions or found that some lesions that were not actually there.
Got it. So
yeah, so on the slide that you see, we would have the first time period for the scan, and then in the middle is the second, and on the far left would be the radar segmentations, just to give everybody a visual. But then that's not all the information we have when we're doing this. We have really large amounts of data. So for each person, at one time point, you don't just have one MRI scan, you have 4 different MRI scans. And so what we're showing here is just one slice of the brain, but an entire brain is 180 slices. So this is a lot of data. And each little part of this brain image is a number. So we've got tons of data for each subject. And then each subject, as Ciprian said, came for a couple of visits. So we have, I think, 11 visits from each subject. And then we have multiple subjects as well. So this is a lot of data that we're dealing with. And so when you're dealing with this amount of data, sometimes it's difficult to use really complicated methods. And so one of the things that we tried to do, and that worked out really well, was to use a logistic regression, because you can fit this really fast on this amount of data. And the results you get are like highly interpretable.
Excellent. So some of the complicated methods just chew up a ton of CPU time,
yeah,
right?
No,
and they can basically freeze the server for days if you have enough data, and so they're not very practical either, right? For quick turnaround, etc.
We've been working on it now, and you can fit a logistic regression model on this data in under an hour. And we've tried some more of these like machine learning techniques that are really popular now, so like a random forest or other things like that. And those can take well, we have some running that have been running for weeks.
Is that right? Wow.
Yeah. So
so we'll check back in around
so
Thanksgiving,
maybe,
or whatever.
Maybe next year I'll probably tell you. But we found that even if you leave these running for a week or so, the performance is exactly the same as logistic regression. If you put smart things into your model, you get the same performance out.
Interesting. So tell us about the model. So what is sort of your unit of measurement? Is the voxel itself or the pixel on the brain, and you're sort of indicating whether there's enhanced gray matter there or enhanced your outcome that you sort of dichotomize for this?
So the outcome for this is a map of where there's lesions. So we actually trained on hand-drawn maps from a radiologist. So they'd be like the maps that we showed you in the beginning. There's a 0 if there's no lesion and a 1 if there's a lesion in that voxel. And they are fit on like individual voxels of the brain, so little pixels.
So you have this matrix of voxels for any...
Yeah, it's actually an array though, so it's like
three-dimensional.
Yeah,
right. Okay, sorry. So we're taking it.
So it's like the whole brain.
Most brains are not a square.
That's right.
There's big problems.
I knew I shouldn't have done that low-cost MRI, right?
Yeah, but so we fit the logistic regression model on these voxels in the brain, and then you get out a set of coefficients. So you guys have probably done this in your class. You'll fit that, and you'll get your beta coefficients. And then when a new study comes, you can take these beta coefficients, multiply them by the images, and then you can produce probability maps.
So you get predicted probability maps of lesions. Oh, okay, excellent.
Yeah.
And a lot of the power of these methods comes from the difference between what happens between one visit and another. You can think, for example, imagine that you look to find a small animal in an area. And if the animal doesn't move, it's very hard to see. But if you just keep on looking and the animal moves, then all of a sudden you focus on that. So the same idea happens with lesions, because you have the scenery, and then you look at the scenery again, and then something changed. Well, that's the type of thing that we are focusing on, and that's what's helping us. One thing that Elizabeth did not say, but it's something that she's doing right now, is that even the 1 hour thing, we are now reducing it to minutes and actually seconds. Because there is another idea that's very powerful in statistics, and that is sampling, or polling. We do a lot of polling in politics. Well, we can do the same thing in
so you treat your full data set like the population, and then randomly sample from that to get better speeds.
Well, not exactly randomly.
Well, I'm sorry, but some sort of sampling. Intelligently sample. Right.
So it's a little bit more stratified sample, where you look for regions that are likely to be. You
condition on the likely. Got it. I see. So and then from those regions you draw randomly.
Exactly.
Right. Excellent.
So these are the two big ideas that are extremely powerful. One is the logistic regression on hyper big data sets, and the second one is once you understand the problem, you can then go back and actually stratify and do everything extremely fast on very large data sets.
Excellent. And so you actually have an example of one of those probability maps, right? Got it. So can you
Yes.
walk us through this? You know, we're going to show it up on the.
Oh yeah.
Yeah. So it's a
the,
really cool graph.
So the probability map, this is what you can make with those coefficients from the logistic regression. And on this particular map, we've colored areas with a higher probability of being a lesion as red, and areas with a lower probability of being a lesion as blue. And so you can see from this image we have on the top there'll be the images from the baseline, and then the next line are images from the follow-up visit. And so you can see there's a new lesion. I believe it's circled with a red box or squared with a red box. And this probability map was able to find that. Or you can see that the lesion's been detected.
The probability is a lot higher.
The probability is much higher in that area. And you can take these probability maps and you can threshold them to kind of create these binary segmentations that are just like what the radiologist gave out. Because if you give this probability map back to a clinician, they're probably not going to be too happy with you because
they don't like the different ranges of certainty, right? They want to know
this
yes
is
or
this
no,
and this is not this. So you want to give, you want to kind of produce something that your collaborators want to use. So.
Excellent. And speaking of that, this is a nice segue into what John's doing with actually the visualization of this. And you just clearly you've heard about how many layers of this data are and how much information is in these data sets. And so simple exploratory data analysis is not necessarily so simple when you have elements of time, different slices, and et cetera across different people coming together. So John, tell us a little bit about how you've worked to make this manageable.
So for the example Ciprian was giving earlier about the animal in the woods, you know, where you're looking multiple times and you're trying to find something. That's not always so easy if you take the same slice over time, right? Okay. So the same slice is supposed to be representing the same part of the brain over different time points. And so when you try to match up two separate images, it's not always perfect. It's usually very good, but it's not always perfect. So it's not so easy to see sometimes when they don't overlap 100%. And also it's not so easy to see necessarily where something lights up that could be small on this slice, and if you went to this slice down, maybe it's a lot easier. So what we thought is in 3 dimensions it would be a lot easier to see these things, right? So pretty much we've taken predicted probabilities from Elizabeth's algorithm and had them over time. So what would be represented is a 3-dimensional brain with lesions over time. So it shows you at visit 1, these are the lesions predicted by Sublime, and time 2, these are the lesions. So you can see whether they're kind of jumping around a little bit in the sense that maybe the overlay is not 100% accurate, so that your brain here might just be moved somewhat slightly. So that when you try to actually overlay time 2 and time 1 brains, even though it's your brain does have a little bit of morphology to it.
Sure.
So overlaying them isn't always perfect. So we can potentially see within a subject over time how well that matching up kind of does. Okay. And also you can see the progression of lesions on a whole brain level, right? So you see a whole brain image, and then you see the entire lesion set of lesions. And then you go over time, you see, oh, well, we see down here a lesion was enhancing down here. And then over time, you see it either gets bigger or maybe it kind of gets a little bit smaller or something like that. So you can kind of get a full representation of the brain, because inherently it is 4 dimensions, right? It is a 3-dimensional brain over time.
Right.
And so
time, the 4th dimension, yeah.
And so we've also tried to adapt this to stroke because, you're trying to get a blood clot out of the brain. So really you want to see where is that actually going away, right? So which areas are clearing faster than others, because that could potentially show you or indicate if they're going to do good functionally later down the line. So if you're taking blood out of areas that are pushing on maybe some function, some motor function, or verbal or reading comprehension or something like that, and if you get those out quicker, maybe that person does better. So looking at this at a comprehensive 4-dimensional level is kind of where we're at. So what we've not designed, but implemented for our data is the ability to have this online, so you can just access it from the web and don't really need to to download and learn like a whole new software, where it just represents the brain in 3 dimensions, and you can click over time to see how the progression is, and you can move it in 3 dimensions, so you can see the back of the brain, the front of the brain, Whereas in other examples, you see the slices over time, but that's problematic because, like Elizabeth said, there's 180 of those slices, right?
So trying to look back and forth.
Yeah. And then you add 10, 20 time points onto it, you're going to be sitting there for a while to see an overall kind of message.
It makes the Rubik's Cube look easy, right?
Yeah, exactly.
So like,
and don't mix up the slices then.
That's right, exactly. So we'll put a link to this on. It's part of the description on the video, so you can see this in action. It's really cool.
Yeah. So John is been leading this effort on 4D visualization, which is another very important component of exploratory, but also analysis of the data.
Well,
so
sure.
We want to incorporate this type of work with the analysis in a way that's actually helping the analysis, because none of the analysis that we are doing is the end game.
Right. It's all interim steps too.
There are many interim steps, and then there is a lot of back and forth between the data and the images and the results. And then you want to see, if you find something, where is it in the brain located and why? Is it just an artifact, or is it something that was really there?
Yeah. And on top of that, we have really been focused on usability for like an end user, for like
researcher,
clinician.
Mm-hmm.
Yeah, and also talking about population-based inference, right? So if I show you one person, this is where their lesions are. That's good. If I show you another person, okay. What we've been discussing is how to kind of represent that on a population level, saying, within the average brain of this population, where do the lesions happen, right? And so if you kind of can describe a population like that, then inherently you want it kind of in 3 dimensions, right? You want to show a brain, right? I mean, you can show individual slices of probability maps on a population level, but a full brain map kind of tries to give you the full picture for that population.
Yeah. Excellent.
So population inference is one added hopefully gain that you can.
So not only the estimation piece, your descriptive statistic, but the uncertainty in it, right?
Yeah.
Yeah, and that can also help if you think about, so that's kind of the cross-sectional idea. Where are lesions?
Right.
But if you look at, say, 1,000 subjects who have MS, and suppose that all of them were just diagnosed with MS, and you look at them over time, say monthly, are certain locations more likely to have lesions than others? And how is that over time? The fact that you have a lesion there will lead to something else in a different part of the brain later. And this is something that it's very hard to do unless we have the visualization tools and the analytic tools to go back and forth between the two.
Excellent.
Exactly. So you can envision a natural history of disease where you kind of bin people on how long they've kind of been diagnosed with MS and see that how that 3-dimensional kind of histogram changes over time, right? So as the disease progresses, does it really target the back of the brain, this part of the brain, or something like that? So you can kind of see 4-dimensional population-based estimates, right? So
awesome.
Yeah.
All right. Well, this has been, I've learned an incredible amount, and I already knew something about this going in. So hopefully you guys really, I appreciate it as much as I do. I'm sure you do, the willingness of all 3 of them to come out and talk to us about, share their work, share their images. And of course, we'll have a link to the SMART website so that you can see more about this if you're interested. So again, thank you guys so much.
Thank you, John.
We'll see you on the next episode.